Rachel the science gal, Rachel the science gal. Rach, Rachel the science gal. Hi there, 4-Hers and friends, it's Rachel here, and we're back again for another week of 4-H related activities. This week is all about forensic science or spy science, and today we're going to be looking at extracting cheek DNA. So for today, I have my trusty lab book. Very important to always follow instructions, or as we call it in science, procedure, when we go about doing a science experiment. So let's just talk about DNA. DNA contains the instructions for making an organism, including you. Your DNA determines how you look, what blood type you have, even your tendency to get some diseases. Almost every cell in your body contains the same DNA and same genes. Some cells, such as gametes, have half as much DNA, and mature red blood cells have none. Each chromosome is made of a single long strand of DNA. So in this activity, we're going to isolate our very own DNA from our cheek cells. First, we're going to break away the membrane surrounding the cells in nuclei, and then we'll precipitate the DNA so you'll be able to see your DNA. So for today's activity, you're going to need salt water, a small cup, a 25% soap solution, and we'll go through how to make that, and ice cold rubbing alcohol, and also some different measuring spoons. So let's go ahead and start by making our soapy solution. It is 3 teaspoons of water and 1 teaspoon of soap or shampoo. So this makes it 25% because 1 out of 4 pieces of this is soap and 3 out of 4 is water. So we'll give that a little bit of a stir. And I did use warm water because water is usually what helps make bubbles. All right. Then I need to make my salt water, and it is a half a teaspoon and 8 ounces. I have already pre-measured my 8 ounces through my Keurig, but if you need to, just use a measuring cup. And I did use warm water because that's what helps salt dissolve. So you need half a teaspoon. I have a 1/4 of a teaspoon right here. So you'd need 2 to make a half. We're going to mix that up and let it dissolve. So what you're going to want to do is you're going to want to swish 2 teaspoons, so about 10 milliliters of salt water from, I'm going to put 2 teaspoons in here. And your goal is to slough off as many cheek cells as possible. And you should time yourself for 30 seconds. I would suggest having several different types of or measuring spoons, just so that you aren't cross-contaminating. So I need to do, that is a teaspoon, 2 teaspoons of this saltwater solution. And then I'm going to swish them in my mouth vigorously for 30 seconds. So I'm going to set my timer right now. All right, then you're going to spit your water with the cheek cells back into the small cup. Then you're going to want to take a teaspoon of the soapy solution. So this is a half teaspoon. Remember what I said, I don't want to cross-contaminate. So 2 half teaspoons equals a teaspoon of our soapy solution. And we are going to pour your cheek cell water solution into your soapy solution. So then we are going to gently mix the solution for 2 to 3 minutes. Try to avoid creating too many bubbles. So the soapy solution breaks down the cell membranes that are made up of fats, just like soap breaks down grease on your dishes. We're gonna go 2 minutes and we'll check back in. All right, so last part of this is that you are going to want to take ice cold 95% ethanol or ethyl rubbing alcohol. 95% can be kind of hard to find. I actually had to buy this on Amazon, but 91% you can find in stores. I know some people were probably buying it before the whole COVID-19 stay-at-home order was enacted. So you might have 91%. If you have 70%, go ahead and try it. We'll see if it works. But what you're going to want to do is take 2 to 3 teaspoons, and I'm here alone, so I'm actually going to measure out 2 teaspoons first. And you want this to be ice cold. This has been sitting in my freezer since I got it, and you want to keep it as cold as possible. So let's give this one final little spin. All right, and then you're going to want to tilt your soapy, cheeky solution and pour this down the side of your tube. You don't want to mix it. We're going to let it stand for about 1 to 2 minutes, and we'll see what happens. All right, welcome back. So we've let this stand for about 2 minutes, and there's a little bit of clumping going on. It's going to be really hard to show on the camera, but if you look at it from the side, you can start to see white clumping going on, and that's actually your DNA. Well, it's my DNA. So research laboratories actually use a similar procedure to isolate and study DNA from different organisms. So give this a try at home. Let us know how it goes. I can really start to see some clumping right now, but it is really hard for the camera to pick up. So give this a try, let us know how it goes, and let us know what you learned by doing this activity. Is there something that you didn't know before, and maybe this kind of sparked an interest in maybe forensic science? Or is it something that you've known all about all along? Let us know in the comments below. All right, have a wonderful day. Bye.